Welcome to my presentation. My name is Melda ESKİTOROS. I am an associate professor at the University of Health Science. The topic of the presentation is the development of hyaluronic acid-coated curcumin-loaded polycaprolactone membranes for biofunctional wound dressing applications. In the content of my presentation, firstly, I will give a brief information about skin infections, and wound dressing materials. Then I will explain the specific properties of the polymer, hyaluronic acid, and model drug. After that, I will talk about the production technique, electrospinning. After mentioning the objective of the study, I will describe the experimental part. The last part of my presentation, I will give you the results, and finally, I conclude my presentation. The skin is the largest organ in the human body by weight and surface area. It is made up of several different substances, including water, protein, lipids, minerals, and chemicals. The top layer of the skin, the epidermis, which consists of keratinocytes, is critical for survival. Under the epidermis, the dermis is a thicker layer of the connective tissue that consists of extracellular matrix, which give mechanical strength and elasticity. However, the structure and functions performed by this organ can be affected by cuts, burns, surgical incisions, or illnesses such as diabetes. The wound healing process begins immediately after the wound is created and consists of four sequential and simultaneous stages. First one is the hemostasis, second one is the inflammation, proliferation, and last one is the remodeling. In addition, traditional dressings can cause dehydration of the wound, and increased adhesion can also cause discomfort and pain to the patient and delay wound healing. The most widely used wound dressings in clinical applications include traditional materials, such as cotton or bandages. These dressings are cost-effective and have good absorbency. However, traditional wound dressings simply isolate the wound from contaminants, as they passively function in the wound healing processes. These drawbacks have stimulated the interest in new types of wound dressings. With the development of technology, there are various types of dressings for different wounds, and choosing specific materials for the wound is important for better wound healing. The main dressings commonly available on the market today are films, foams, sponges, hydrogels, and fiber films. Among these materials, electrospinning nanofiber membranes stand out as having great potential for wound dressings. Electrospinning is a simple, versatile, and cost-effective method to produce nanofibrous membranes. In addition, bioactive molecules have also been incorporated into the electrospun fibers to improve the biological performance of these membranes. Curcumin is a natural compound with a high potential wound healing due to its anti-inflammatory and antioxidant properties. It can increase epithelial regeneration, fibroblast proliferation, and vascular density, which has an accelerating effect on wound healing. Because of poor aqueous solubility of curcumin, the bioavailability and sequentially the therapeutic efficiency of curcumin are restricted. Thus, the innovative, efficient delivery systems are needed to ensure curcumin solubility and stability characteristics besides improvement of pharmacological potentials. PCl, polycaprolactone, is a biocompatible and biodegradable polymer that can be electrospun easily at low voltages and can provide the required scaffold mechanical resistance in aqueous environments. It was approved by U.S. Food and Drug Administration, the most used polymer due to its good mechanical, biological, and physical properties. However, PCl is highly hydrophobic. Hyaluronic acid plays a crucial role in numerous biological processes and can exert positive effects on the wound healing processes. The low concentration of the hyaluronic acid solution also have high viscosity, which makes the electrospinning of hyaluronic acid challenges. Hyaluronic acid is an essential component of the extracellular matrix. Therefore, the aim of the study is to develop a multifunctional nanofibrous wound dressing by integrating curcumin-loaded PCL fibers with hyaluronic acid surface modification to enhance bioactivity, hydrophilicity, and overall wound healing performance. For the fabrication of wound dressings, first the PCL was dissolved in the chloroform dimethylformamide solution under constant stirring for 24 hours at room temperature to ensure complete dissolution. 5% of curcumin with respect to the polymer amount was added to the polymer solution. The parameters of electrospinning system were shown in this slide. Then the electrospoon membrane was deep coated in the 1% of hyaluronic acid solution for 72 hours at 37 degrees Celsius. The first results of this study is the SEM analysis. One of the most significant parameters for electrospoon membrane characterization is the surface morphology of the materials. As shown in the slide, the membranes were obtained smooth, beadless, and random morphology. The membranes have been successfully fabricated by this method. The average diameter of the fibers was calculated and given in the table. As you can see, the incorporation of curcumin into the PCL nanofibers decreased the average fiber diameter. This is probably due to the change in viscosity of the PCL solution. However, after the deep coating processes, the average fiber diameter was significantly increased, as obtained by Basmande et al. They reported that fibers widening were observed for hyaluronic acid-coated nanofibers. For atomic force microscopy analysis revealed that the incorporation of curcumin into the PCL fibers increased the average surface roughness. This enhancement can be attributed to curcumin's heterogeneous distribution and partial aggregation on the fiber's surfaces. The increase in roughness is expected to improve cell attachment and proliferation, potentially enhancing the functional performance of the wound dressings. FDR analyses confirmed the successful integration of hyaluronic acid and curcumin into the PCL fibers. The spectra of the PCL curcumin hyaluronic acid membrane exhibited characteristic peaks of PCL, hyaluronic acid, and curcumin, indicating that both bioactive agents were incorporated without altering the polymer's chemical structure. For water contact angle measurements, these results show that pure PCL and PCL curcumin membranes exhibited high hydrophobicity. In contrast, the incorporation of hyaluronic acid into the PCL curcumin membrane significantly reduced the contact angle, indicating enhanced hydrophilicity due to the hydrophilic functional groups of hyaluronic acid, which can promote better cell adhesion and proliferation. To sum up, SEM analyses show that curcumin incorporation reduced fiber diameter due to decreased solution viscosity, while hyaluronic acid tip coating increased the average fiber diameter. Atomic force microscopy analyses reveal an increase in surface roughness after curcumin addition, attributed to heterogeneous distribution and partial aggregation on fiber surfaces. FDR spectra confirmed the successful integration of both hyaluronic acid and curcumin into the PCL matrix without altering the polymer's chemical structure. And the last one is the water contact angle measurements demonstrated that hyaluronic acid incorporation reduced hydrophobicity, enhancing membrane hydrophilicity, and potentially improving cell adhesion. Therefore, the developed hyaluronic acid-coated curcumin-loaded PCL membranes demonstrate improved biofunctionality, indicating strong potential for advanced wound dressing applications. Thank you for your listening.